All right. So, new way to do class. I don't know what I'm doing. Maybe you know what you're doing. It's all new to me. I don't do YouTube videos. I mean, there's one with me with a hood on and playing the guitar, but don't worry about that. Anyway, so this is Brett Rodriguez. You know, I'm your anatomy professor, physiology professor, and I'm also a pediatrician. So we're here in my pediatric office trying to do school because you're hunkered down, hopefully, trying to avoid getting coronavirus. After a month ago, I told you there was nothing to worry about. Here we are in, you know, some kind of terrible dystopian novel, trying to figure out how we're going to get through this semester. I know it seems like forever ago since we had class. It seems that way to me too. We are going to get this done. We're going to rush through muscle today. We're going to get this done. A big part of this lecture is watching videos. Well, you have my slides. You don't need me to sit here and go and watch the video with you. You can just do the slides on your own. So look, what I would do if I were you is I'd pull up the slides and I'd follow the slides while I'm doing this lecture. And if you want, when it's over, you can re-watch the videos. These videos are really good. Watch them a couple of times. And you know, I told y'all that A&P crash course that John Green does is really good. The one on muscles is really good. Okay? So watch that video. It's easy to find on YouTube. You got nothing else to do, right? Turn off Netflix for a little while, watch YouTube. So we are going to do muscles now. All right, you know there's 700 skeletal muscles. There's a whole bunch, but we're not just going to talk about skeletal muscles. We're going to talk about smooth muscles and cardiac muscles too. But right now we're talking about skeletal muscles. It's composed half of your adult weight. You know, kids, it's a little less probably, but so this maintains your posture. It allows you to move. You couldn't move at all without muscles. Protection and support. Heat production. When you shiver, that's generating heat. Your muscles do that. If you exercise, you will notice you will get hot. That's heat production because you're burning lots of calories, burning ATP, that generates heat. All right, and also, you know, your movement of things through the body, some of that happens through skeletal muscles, like when you chew, for instance. That would be movement of materials. Okay, so we're going to talk a little bit about skeletal muscles. This is the formation of skeletal muscles from myoblasts. See how they start off, these individual muscles, but then they stretch out and they make these long multinucleated cells. Is important. Most cells that we have studied so far didn't have multiple nuclei. Skeletal muscles do. They have lots of mitochondria. Look at that, 300 plus mitochondria. That's a big deal, right? You're using up lots of ATP when you run, when you exercise, when you do stuff. So you need lots of mitochondria to make that ATP. And we're going to see how ATP is utilized to make muscles contract before this lecture's over with. So here's just a short description of these three different types of muscle fibers. Every muscle has some of these fibers in them. Depending which muscle you're talking about, some have more than others. So we have these slow oxidative ones, and then these fast oxidative ones, and these fast glycolytic ones. Okay, so this would be, you know, short duration, intense movement would be these fast glycolytic ones, like the ones you use to sprint, or that you'd push heavy weight with. Okay, then you also have slow ones that you use when you're maintaining your posture. These are the ones that are good at contracting for long periods of time over and over. And then these are in the middle. Okay, that's really all I'm going to say about that. So let's talk about muscle anatomy. This must be one of the bicep muscles, I can tell, because this is clearly the head of a humerus. And that tendon's fitting between that little space right here. So we see, you have to know this anatomy. There's a tendon. Muscles come down to a sheet of fascia, turn into a tendon. The tendon attaches to a bone. You have to know that the tendon is what attaches to a bone. The tendon is what the muscle fascia turns into. So you have your deep fascia, and then the layer over the whole muscle group is the epimysium. And then these little individual bundles here are called fascicles. All right. The lining inside around the individual groups is the endomysium. And here's a single muscle cell. You can see the satellite cell there, and then the nuclei, multiple nuclei. Look at the slide. I mean, it's like always with anatomy, you just got to learn it. All right, so here's the microscopic anatomy of a muscle cell. Now, you know, we always got to come up with fancy names for things. So in muscles have fancy names. The outside coating of a muscle cell, right, instead of calling it, you know, the plasma membrane, it's the sarcolemma. Sarcolemma. Sarc is just Greek for flesh. So the sarcolemma, that's the covering there. And then the inside, what we normally call the cytoplasm in muscles, we call it the sarcoplasm. All right, so we have these fancy things called T-tubules, and we will get into detail on what T-tubules are. It's actually an extension of the sarcolemma, the plasma membrane, that goes inside the cell all through the cell, the muscle fibers. And the reason for that is so that the outside can get to the inside very quickly. That's the point of the T-tubule system. It's a little confusing when they just start showing you pictures of T-tubule. Well, what's the point? The point is to be able to get the electron changes, no, not the electron changes, sorry, the electrolyte changes inside the muscle very rapidly. Okay? So what you see here, this is the interstitial fluid. This is outside of the cell. Okay, and here's the inside of the cell. Notice there's a voltage difference from the inside and the outside. Okay, it's negative inside, positive outside. If you had a little tiny voltmeter that you could put on one side and the other, it would be minus 90. Would be the millivolt difference between the inside and the outside. And that's going to become significant when we get to nerves. We're not at nerves yet, and I hate that because there's no way to talk about muscles without talking about nerves. But we're going to see how that voltage change happens and what it does. In this case of the muscles, later we'll learn what it does to the nerves. So see out here, what's all this stuff floating out here? Okay, this is electrolytes, this is sodium, and inside is potassium, right? We know that there's more sodium outside than inside, and there's more potassium inside than outside. So what happens is, as these channels change, as they open, sodium comes in and makes this more positive, and the positive change inside is what opens these channels, which go into the sarcoplasmic reticulum. Remember, we said everything has a fancy name. Your endoplasmic reticulum in the rest of the cells, and muscles is the sarcoplasmic reticulum. So what happens when the voltage changes is the calcium comes out, and then the calcium is what actually makes muscle contraction happen. And we'll go into details on that in a second. Okay, so this is the triad. Here's calcium being held inside the sarcoplasmic reticulum. It's going to be released out here into the cytosol, right? The sarcoplasm, to make contraction happen when we get to a contraction. All right. Each muscle fiber is composed of thick and thin filaments. You have to know that. So thin filaments are tropomyosin, actin, and troponin. Thick filaments are the myosin, right? The thick filament has these myosin heads, and they're just called that because of the way they look under electron microscope. All right. So we see how myosin, here's an electron microscope picture of myosin. Notice you can actually see these little heads. This is a cartoon of how they look. You can look at your slides and see that. They have an ATPase because they can break down ATP. And we're going to see that they have a site to bind with actin. The binding between myosin and actin is what actually causes contraction. So here's a thin filament. It's made up of actin with troponin and tropomyosin around the actin. We're going to see how that works in just a second. And they also have an active site ready to bind with myosin, but most of the time the active site is covered up by these thin filaments right here. Okay. The individual contractile unit of a muscle is called the sarcomere. So every muscle fiber has numerous sarcomeres in a row. The sarcomeres is what shorten. When the sarcomeres shorten, and they all shorten, that's what actually causes a muscle to shorten. You know, this muscle, your bicep, only works if you're pulling. It doesn't do anything when you're pushing. There's no strength in a muscle pushing itself out. It doesn't lengthen out. It's either contracting or it's relaxed. It doesn't push. What makes this hand go down is your tricep, pushing it down. And it's the same thing with the way your legs work. Your quadricep only pulls your toe up, right? It pulls it up like that. It also does a little bit to pull your leg at the hip, but it only contracts. They don't push. See, they have the sarcomere divided up into these sections, and this is anatomy that you need to know something about, but you don't have to be an anatomist. You don't have to know this perfectly. Okay, so this is a video on the sliding filament theory. We're not going to watch it now. I want you to watch it on your own, and I really want you to watch it. But you see in the picture, here's an electron microscope, how the sarcomere, because the sarcomere is from one Z disc to the next, how as that thing shortens up, that's the actin and the myosin working together to get shorter. That's what causes a muscle contraction. Okay, so when does a muscle know that it needs to contract? Well, for skeletal muscles, it's when the brain tells it to, right? And the brain sends a peripheral nerve down, and it joins. This is a nerve joining with the muscle fiber at what we call the neuromuscular junction. And this synaptic knob here is the very end where the nerve actually meets the muscle. And there's these wonderful little vesicles full of a specific neurotransmitter called acetylcholine. I'm going to just draw this over here. Becky, you're pretty sure that they can see the board there? Yeah, she says we can. So this is what happens. This is my neuromuscular junction, all right? Here's my muscle. Here's my nerve bulb. See, the nerve synapse meets on there. And already, before an impulse from the brain gets here, there are little vesicles full of acetylcholine. We're going to abbreviate that ACH. Ready to go. They're just sitting there ready to go. And when that voltage comes down, there's a change. What happens is these voltage-gated calcium channels open up, and calcium rushes in. Calcium binds with these vesicles. The vesicles move down here. You know, they're moving down those little chains. They don't just move just floating around. They're being moved there. And then through exocytosis, acetylcholine is spit into this space. And then there's some acetylcholine sodium channels that when the acetylcholine binds to it, we call them ligand gated, because it's not a voltage change that makes them open, it's the ligand. Remember, the ligand was what we called anything that binds with a protein that makes it change shape. So when that happens, that changes the voltage, because now sodium can go in here. So sodium comes in, and sodium opens more because of the change. You know, this gets less negative. It doesn't get all, you know, just a little bit, just gets a little bit positive, but it certainly gets less negative. And once that happens, these start opening, and now more sodium comes in. And the sodium coming in is what causes the calcium to come out of the sarcoplasmic reticulum. Like I said, the videos explain that better than I can, but that's the summary. You're going to have to be able to reproduce this for me for the test. You're going to have to know how to do that. Okay, so this is the first video that goes into detail about the neuromuscular junction, and it's kind of what I just explained. And then we get into detail on how the actin and the myosin bind to move things. All right. So this slide is a pretty good summary of everything that happens. So I'm going to review this. I still expect you to know this on your own. I'm not going to take up a lot of time because I know watching videos like this can be kind of boring. But we're just going to review everything we've talked about so far. So here we see this nerve signal comes down. We call that an action potential. That's our fancy name for it. Nerve signal comes down, opens these voltage-gated calcium channels. Calcium goes in. Calcium binds with these vesicles of acetylcholine. That goes into this, it gets exocytosed into the space. Acetylcholine opens these sodium channels, and then that change in voltage causes an action potential all along the muscle because of the change in voltage. And the change in voltage all along, see, it gets to come down through the T-tubules that we mentioned earlier. That opens the gates on the sarcoplasmic reticulum, letting calcium out of the sarcoplasmic reticulum into where all those muscle fibers are. And then the binding of the calcium from here with the troponin moves the tropomyosin out of the way, right? It binds with troponin and moves the tropomyosin out of the way, sorry. And then actin and myosin can get together and cause that cross bridge formation that causes the contraction. Okay? So now we're going to talk about how is it that what we just learned about can actually equal nice smooth motion like you see in the animal kingdom, right? All animals have these beautiful smooth movements, sleek. Humans have nice smooth movements. How can that herky-jerky stuff we just learned about, how can that equal smooth movements? And the answer is because you have graded contractions. Not all the muscle units contract at the same time, depending on how long it takes for the nerve signal to get there, and different motor units contract at different times. It makes for one nice smooth movement. And so what we've learned through experiments we've done over the years, we know that they either are contracting or they're not. So why isn't every movement jerky? And the answer is, I'm skipping ahead because I want to answer my own question. As the neurotransmitter nerve signals get there, the muscles can be increased in what we call wave summation until you finally get to where the muscle's completely tight. And this will stay that way as long as it keeps being called upon to contract. In this case, somebody did an experiment with voltage, but it works with when the nerve's working on it too. And then finally the muscle gets fatigued, it can't keep that up. Okay, so we need to learn the names of these isotonic, concentric, and eccentric contractions, and it just has to do with what kind of contraction you're having. So if you're just holding force against something, but you're not moving, you're just holding it, that's isotonic. If the muscle's shortening, that's concentric. If it's lengthening but still holding some pressure, that's eccentric. Okay? And then if you hold something just not moving, that's isometric. All right. So we're going to briefly talk about metabolism in a skeletal muscle. We know that ATP is what causes the power stroke. And so where do you get that ATP from? Well, you can generate it in the mitochondria like we've seen, but we also have creatinine kinase is all the time holding a phosphorus that it's ready to give for whenever you need to do a quick sprint. I'm not going to go into great detail on that. You just need to know that there's more than one mechanism for regenerating your ATP in the muscle cell. So the first few seconds of exercise, you have that phosphagen system where creatinine phosphate gives its phosphate to the ADP so you can have the ATP. It rapidly regenerates it. Then as it goes on, you use glycolysis, but as you know, glycolysis only gives you 2 ATP, so if you're in an oxygen depleted environment, you only get 2 instead of the 30 you'd get if you had lots of time. So in anaerobic, without oxygen, you end up with just this 45 seconds of moderate exercise. You just get glycolysis with lactic production, and there's an oxygen debt that has to be paid back later. Okay, after 2 minutes, the only movement you get is from aerobic respiration because you've used up all your short-term ATP, and then of course that's how you get the majority of your ATP over time, is from the whole cycle within the Krebs cycle and in the mitochondria. But you have to have oxygen to do that. So the question is, what causes fatigue? Okay, so there's a psychological component to it. The muscle can also run out of either ATP or the right balance of sodium and calcium to make the thing happen. And your nerves eventually even can quit having enough ACT to be released into the space, acetylcholine, to make contraction happen. So that's these three components of it: psychological, what they call muscular, where you don't have enough either electrolytes or ATP to make it happen, or maybe you've run out of sugar. And then we have these synaptic vesicles can run out of acetylcholine. All right. So very briefly, what's the difference between how cardiac muscle works and how skeletal muscle works? Okay, not very much. Cardiac muscle works very similarly to skeletal muscle, but you don't have any control over it, right? Thank goodness your heart just beats without you having to think about it. There are sarcomeres. That's why it's striated. When you look at it, it looks striated under a microscope. But it also has pacemaker cells throughout the heart, so that the heart has a natural contraction to it. Even if you cut off nerve supply to the heart, it'll still beat, like every 30 seconds or so, just on its own, because it has its own natural pacemaker cells. We're not going to go into that. Okay, smooth muscle. Smooth muscle is a completely different action. It's not striated, and it has a different electrochemical basis for what causes the smooth muscle to contract. Notice we have these dense bodies all through the cell, so that when the cell contracts, it's because they have these crosses of actin and myosin, and that's how the whole thing just gets shorter. The whole cell gets shorter. Notice it's not attached on each end the way skeletal muscle is. They're all bunched together, and as they bunch up, whatever the thing is that is being contracted gets smaller. So in your aorta, when your aorta contracts to get smaller, there's smooth muscle there that's making that happen. When they relax, it gets bigger. Okay, for a woman having a child, when her womb contracts, it gets thicker and thicker as those muscles bunch up more and more. They get more in this position and less of this relaxed position. Okay, so there's no sarcomeres, and there's also no troponin or tropomyosin. Okay, there's this excitation contraction coupling mechanism with calmodulin. You need to know calmodulin's involved. You need to know that this myosin light chain kinase is involved. That's really all you need to know. You need to know that they're a latch bridge formation. Okay. So we're going to talk about the different smooth muscle types. There's multi-unit ones in the autonomic nervous system, and then there's single unit ones. These are myogenic. Example would be in the GI tract. That's really all you need to know about this from my class. Okay? And so next class we'll talk about something else. You need to be doing your homework. Please do your homework. It's clear from the way the semester's gone so far that some of you were really interested and concerned doing this class well. If you're that person, you're going to do great. If it's obvious to me that you haven't cared up to this point and you're still not doing your homework, I'm not going to have any sympathy for the fact that the world's falling apart, okay? If you're not doing your homework and you're not trying, don't expect just to get an A in this class just because things have been terrible. I just want to make that clear. You still got to try. I know there's a lot of mess going on right now, but we got to get through it. I got to get through it. You got to get through it. That's it.